I'm Roger Buick, and it's my pleasure to introduce Professor Anitra Ingalls from University of Washington School of Oceanography this week. Many of you will know Anitra already, but for those of you who don't, she got her PhD at Stony Brook. She then did a postdoc at Harvard, and she's been at University of Washington since 2004. She works mostly in the area of, I guess you could broadly describe it as marine biogeochemistry. She published a great deal, including perhaps, well, many of her most highly cited papers as part of the team that was the first to discover the marine ammonia oxidizing archaea, along with Dave Stahl, for instance, who's also in the astrobiology program. For those of you who are grad students and looking for a mentor right now for maybe a research rotation project, I should point out that Anitra currently has millions of dollars in grant funding from the Simons Foundation. And I'm sure that you could find a really good research rotation project with her, because she's bought some fancy new equipment recently, and I'm sure is looking for users as well. So she'll be talking to us today about the metabolic products of phytoplankton, in particular organic metabolic products. And this has relevance not just to the Earth and its oceans, but to the potential range of chemical compounds that you might find in an inhabited ocean in an icy world. So it's a talk about actual and potential biomarkers for life on both Earth and other planets. So Anitra, please take it away.
Thank you, Roger. I will share my screen. Here we go. And I will start my talk. And okay, I will assume everyone can hear me, or else Roger would tell me. So yeah, today I wanted to talk to you about, well, what I've titled my talk, A Mass Spectrometer's View of Microbial Life in Ocean Worlds. It's a bit fantastical because I work in only one ocean world, not many ocean worlds, but I think that what I'm going to tell you about applies to other ocean worlds potentially, and that's what I'm here for. I've looked kind of from afar for a long time at the astrobiology program, wanting to get more involved, and just it's always hard to find the time and the hook. And I now have the hook because I have a student, Josh Sachs, who is working in my group, and he's doing the dual degree program. So now here I am, becoming part of the group finally. And here is my research group. I like to introduce them. I have the best research group ever, anywhere. So I'm really happy to have them all in my group, and I want to just introduce a couple of them. The whole group is really involved in everything I'm going to tell you about. They're very collaborative. Most of the members of the group have helped somebody to do something and that I'm going to tell you about today, so they're all deserving credit. I'm going to specifically focus on projects by Catherine Heel, Angie Boysen, and Josh Sachs. And my lab manager is Laura Carlson. She holds the whole place together. And my data manager is Regina Leinhart, and she also holds the data together. But everybody has contributed a lot. And I'm going to try to give you a bit of an overview of the kinds of things that we do. So I'm going to touch on a bunch of different projects, and hopefully it won't be too serious. This was obviously from a while ago when we were dangerously close to each other outside at a party at my house. This is a more recent picture of my lab group. Some members have come and gone, and new ones have arrived. So these are the people that are behind the work I'm going to show you today. So ocean worlds. I don't know a lot about them, as I just said, but I do know that there are several in our solar system. And Earth is the biggest one with not the most water. So there's other planets that are much, or other ocean worlds that are much smaller, but have more water on them. I know that this one, Enceladus, if I can even pronounce it, is one that people are pretty interested in, among other ones here. So these ocean worlds are places that we would like to explore. And I think there's been some recent papers talking about how important it will be to communicate and collaborate with scientists that study Earth's ocean in order to explore if and when it becomes possible to actually explore these other planets. So I'm here studying Earth's ocean and thinking about organic carbon in our ocean, which is a sign of life. When you see organic carbon, it's a sign of life. And in our ocean, phytoplankton are the main organisms that convert carbon dioxide into organic matter. So they make almost all of the organic matter in the ocean. They make 100 million tons of organic carbon every day. It's a lot of organic carbon. It's the base of the whole marine food web and also the base of the biological pump. Once produced, this organic matter becomes part of a broad ecosystem that can move this organic matter into different directions. And one of the directions that can go is up the food chain. You can make things like fish and whales. The next place it can go is into the microbial loop, where it will be cycled through a variety of different microorganisms that all kind of contribute to and consume this what's called dissolved organic carbon pool, which can be formed when cells lyse and burst open and spill their contents, or when they're grazed. That dissolved organic carbon can be in the form of very labile material that's very sort of yummy to, say, bacteria that are living in the ocean, or it can become part of the semi-labile or refractory carbon pools, which is actually the largest pool of organic carbon in the ocean. All of these pools can be respired back to carbon dioxide, where the carbon dioxide can go back into the atmosphere. But if it escapes that fate, it can be exported to the deep ocean in the form of sinking particulate organic carbon. And this is where the ocean, this is called the ocean's biological pump. It's where the ocean is able to store carbon for long periods of time deep in the water and in the sediments if it makes it that far. Depending on where you are in the ocean, the amount of organic carbon that is exported from the surface and sinks to the deep ocean is different. And so this figure shows you carbon flux throughout the world's oceans. And these warm colors are places where carbon flux is very high. So there's a lot of organic carbon sinking through the water column. The blue areas are areas where there's less organic carbon sinking through the water column and being sequestered in the ocean. So what controls this, the fate of this organic carbon? I've given you this figure, and we in my lab are interested in all the different directions that it's going, including that whether it's exported or not. And so what it turns out that whether there's many factors that determine which direction carbon will go, as you might imagine, there's probably 100 factors that contribute. But one of those factors is the phytoplankton community, who's there and who they're interacting with. And here's an image from a recent paper showing some very specific interactions between phytoplankton or algae and heterotrophic bacteria, for example, and how these organisms are exchanging things like vitamins and growth hormones and osmolytes, in this case DMSP, which then can be processed and released to the atmosphere and has an impact on climate. Things like various other organic substrates, sulfur-containing osmolytes, ammonia, iron, and iron-binding ligands can be exchanged. So there's a whole series of kinds of interactions that people are uncovering between these among the members of this community that are going to control, in part, how much carbon is produced and how much carbon can be stored in the ocean and how much might be available to move up the food chain. So I wanted to start by thinking about what controls where phytoplankton live. There are many factors that can control the niche space of a particular phytoplankton, the area in which it might like to live, the sort of environmental conditions that it wants to have to be successful. And so those are things like shape and size. You can see these different phytoplankton. Some of them are teeny tiny and some of them are very large. They have different kinds of cell walls, silica cell walls, carbonate cell walls, or no mineral cell walls. Some are motile, some are not. They have different kinds of predator-prey interactions and different microbiomes. They might have different nutrient preferences. Some require vitamin B12, some don't, for example. They have very different cellular stoichiometries. So their carbon to nitrogen, phosphorus, iron, and cobalt ratios will vary from one type of phytoplankton to another. So depending on what nutrients are available, one might grow preferably to another. There's also a level of physiological plasticity that organisms have as inherent in their genomes, I guess, that can control how flexible they are in a variable environment and whether they can survive or not. And also they might produce bioactive molecules, like things like toxins that would actually kill other organisms. So these are all things that can control where phytoplankton live and what community members they live with. And I would argue to you today that all of these are in some way related to the chemical composition of the organic matter. And the chemical composition of the organic matter is kind of what I focus on in my research, and I'm going to talk to you about today. So essentially, my group is interested in thinking about phytoplankton communities and their partners or their enemies, the chemical composition and the molecules that are being exchanged or communicated among this community, and how that impacts these biogeochemical cycles. So to do that, we're thinking about measuring chemical structures. We identify a particular chemical structure, measure its concentration, try to think about its biological source, who's making it, what its function is in that cell and in the community, and then also potentially measure rates and biochemical pathways that are involved in this cycling. So that's kind of where my work is aimed at, and what my work is aimed at accomplishing is getting a picture of the chemical basis for these interactions in this ecosystem. If we're thinking about exploring for life in Earth's ocean and in other oceans, we think about biomarkers, and a biomarker is just an organic molecule or any molecule that is specific to a particular group of organism or a particular type of organism or species. And we don't just look at the chemical structure. Sometimes we can look at isotopic fractionation, which biology fractionates isotopes because enzymes generally prefer a lighter isotope to a heavier isotope. We look at chirality because life produces chiral asymmetry. And then we also just look at the chemical structures. So my group is mostly focused on looking at the chemical structures in the ocean to help us learn about the ecology, evolution, and physiology of marine microbes, and also then infer biogeochemical cycles. So I'm interested in the chemical components of life. So what are they? Cells, all cells on Earth are made of macromolecules primarily. That includes things like DNA and RNA, which are the instructions for the cell, proteins and lipids, which are the architecture of the cell, the membranes, catalysis, which is accomplished by proteins within cells, as well as energy storage compounds like lipids and carbohydrates. Those are the main components of the cell. However, there are other components of the cell as well. These are called metabolites. These are small molecules that are the product of metabolism. So some of these arrows here are indicating some of the main pathways of metabolism in all cells, and these are just some random structures that I pulled out to illustrate that there are chemical molecules that are being transformed. So what is metabolism? It's the sum of all chemical reactions in a cell. So that involves things like conversion of energy, building cellular building blocks like amino acids to make proteins, eliminating waste, and producing things like vitamins and hormones and pigments that cells might use for specific purposes. And then also osmolytes are particularly important in the ocean. Osmolytes are molecules that have multiple functions, but one of their main functions is to help a cell deal with stress, and in particular osmotic stress. So living in a salty environment means you have to have some form of ions inside your cell. Otherwise, you would either burst or shrivel up. So metabolism acts through enzyme substrate pairs, and the substrates are these metabolites. And as you can see from this image, many of the metabolites in a cell are small and polar, and many of them have charges. So here's a positive charge, negative charge. So these are small charged molecules that kind of look a little bit like salt. And the fact that they're so much like salt makes them actually very challenging to measure in the environment. So as a result of these analytical challenges, we don't know the structure or abundance of many metabolites within plankton cells and communities. These metabolites don't just exist inside the cell. Sometimes cells have reasons to excrete them into the extracellular environment, and there they become part of the dissolved organic carbon pool. But cells can also burst through viral lysis or through grazing. And so during mortality, these metabolites are released into the dissolved organic carbon pool, which, as I said, is the largest pool of carbon in the ocean. These excretion rates are almost entirely unknown, and the reasons for excretions are almost entirely unknown. Which molecules are excreted, we don't know. But once the molecules are in the dissolved pool, they're much harder to measure, as I said, because of this presence of salt, which behaves a lot like the molecules that we're interested in. So this dissolved organic carbon starts out, we think, as pretty sort of labile. The microbial heterotrophic microbial community would consider it yummy food with high nutrient value and lots of energy. They also could be things like toxins and hormones, which would affect growth rates and that sort of thing. But they can be transformed, we think, into semi-labile and also refractory carbon, which is a very large pool of organic carbon in the ocean, this refractory pool that's sort of constant everywhere. So at the moment, no studies have really comprehensively characterized the organic material in the ocean in a comprehensive way, partly because it's so hard to do. And there are lots of people interested in this, and they're using indirect tools. So there's some studies that have used things like transcripts of transporters on heterotrophic bacterial cells to try to understand what molecules they might be bringing into their cell to consume as either food or as nutrients. And there's also people who use proteomic approaches to look for transporters that might be bringing food into the heterotrophic bacterial cell. Other approaches are to label an organic molecule and feed it to a community and figure out which organisms are using which molecules. So these are all sort of indirect approaches to understanding the organic carbon available to heterotrophic bacteria in the ocean. And it's interesting because these studies have shown that even though metabolites are approximately, something less than 10% of cellular biomass, they seem to make up a large portion of the transporter activity, both in the transcriptome and proteome of heterotrophic bacteria. And so the question becomes, why are they expressing so many transporters for these molecules that are a small, we think are a small portion of the biomass? And you should keep in mind when I say that many transporters are poorly annotated, as are the biosynthetic and catabolic pathways for these molecules. So in my group, we really are interested in looking directly at these molecules, which ones are being produced and how much of them, and whether they're being excreted into the extracellular environment and in what quantity. So how do we do this? We measure particulate intracellular organic molecules, or we call metabolites. First, we take water either from the ocean using this CTD rosette, which these are bottles that collect water from the ocean. And we can filter that water, or maybe we've grown a culture and we can filter the culture onto a filter that has a 0.2 micron size fraction, pore size. And then that biomass is retained on the filter, and then we can extract it using a modified Bland-Dyer extraction, which is really nice because it gives us an aqueous phase with all of those very polar molecules and an organic phase with the non-polar or lipid molecules. We have to add isotope labeled standards in order to quantify any of the compounds. And we do that for a fairly large number of molecules. So then we take our extract and we use liquid chromatography coupled to mass spectrometry to measure the compounds. This means that we do liquid chromatography to separate individual compounds. They get ionized and then they get detected by the mass spectrometer, and they look like a peak in time that elutes with a specific retention time and is associated with a specific mass and intensity of the peak. We take all the peaks and we align them, and then we take our standard peaks and we use them to quantify the known compounds. And then if there's anything, or there are always many compounds, we don't know their identity, and we use databases to try to identify those compounds. Usually we detect several hundred compounds. 3 to 4 are high quality peaks that we think we actually are metabolites that are meaningful. And we quantify about 100 of those with standards. Even though we have more standards, we don't always see all the molecules we have standards for. I want to point out something about the chromatography that we're using that's kind of important. Traditionally, oceanographers use what's called reversed phase chromatography, which is a way of analyzing relatively nonpolar compounds. In this kind of chromatography, the polar compounds would just fly right through this column and not stick to it at all, and then they would come out immediately, and along with salt and probably dozens or hundreds of molecules would come out right at the very beginning of this chromatogram, which makes it impossible to quantify those compounds. So we have started using what's called Hillick chromatography, or hydrophilic interaction liquid chromatography. And this is a way of separating very polar compounds. And here's an example of 2 compounds, trigonylene and homarine. Don't worry about their structures for now, just names of 2 molecules that happen to have the exact mass. They're isomers of each other, so that we cannot distinguish them using just the mass spectrometer alone. And in reverse phase, they come out at the same exact time in this chromatograph. And they also come out in the very beginning where all the salt and a whole bunch of other compounds are present. So in a traditional approach, you wouldn't be able to tell how much of these compounds were there, or even if they were there at all. And with our approach, they separate and they come out much later in the column, in the chromatogram. So we can actually differentiate them from each other. And we know this one is homerine and this one is trigonylene, and we can quantify them. So we can do this. There's a whole bunch of examples of this I could show you, but I'm focusing on this one in particular. So this is the tool that's allowed us to actually start measuring these molecules for the first time in the ocean. And we're also developing a new tool for doing this, measuring the dissolved polar metabolites. And this is relying on cation exchange chromatography. And my student, Josh Sachs, is responsible for developing, bringing this method to its usable form. And he has started getting data, and I'll show you a little bit of that at the very end if I get to it. So our approach, though, was to start with the intracellular pool, because we decided if you know what's inside the cell, you know what to look for outside the cell. And without knowing what you're looking for, it's hard to tailor a method like cation exchange to actually target those molecules. So with that information of what's inside the cell, we've really been able to make a lot of progress with this dissolved metabolite method. And we're also working on an anion exchange method, but that one's a little bit more challenging because of the way chloride behaves. But we hopefully will get something to work. So I have a lot of projects going on in my lab. I'm not going to tell you about all of these. I'm going to focus on an example from each of these areas. One is to understand the source of metabolites, who's making which ones. And for that, I'm going to show you some environmental metabolomes combined with some metabolomes from a lot of cultures that we've been growing. I'm going to tell you a little bit about physiology and how the organism's sensing of the environment can affect how the metabolome looks. And for that, I'm going to show you some results from a study where we measured metabolites in the environment over a diel cycle. And then finally, I'm hoping to tell you about the fate of a couple of metabolites. So hold on, it's going to be a wild ride. I'm going to start with sources. And to start, I'm going to tell you a little bit about some work that Catherine Heal did. She measured metabolites across a gradient of chlorophyll in the North Pacific. We cruised from, this is Hawaii, if you don't, for the non-oceanographers who aren't used to looking at the ocean, that's Hawaii. And we just cruised north, and we didn't get all the way up to Alaska, but maybe halfway or 2/3 of the way there. And we collected samples every so often and measured their metabolomes. And when we plotted that data in NMDS space, multivariate statistics, it shows that the stations where we collected samples that were further to the north, these cold stations, looked different than the ones in the south, these in the warm water. This is a temperature scale showing the coloring of these data. So we were interested in understanding why, when you cruise from Hawaii into this more northern area where you see an increase in the overall amount of chlorophyll in the water, why don't you just see an increase in metabolites? Instead, you see actually a change in the composition of the metabolomes. And these areas correspond to what we call the North Pacific Transition Zone, which is a zone where you're seeing a real change from the gyre, which is a circulation feature of the ocean that's fairly nutrient poor and warm, into this North Pacific where there's more mixing and colder water. So we can now see that with metabolomes from the environment, we can actually differentiate these two environments from each other. Pretty cool. What's responsible for that? Well, first I'm going to show you a little bit of the data. This is the top 20 metabolites that we can quantify. That means we know exactly how much is there. And this is their mole fraction of each one. So this is showing you sort of the composition of the material, not the absolute amount. And what you can see is that at about 30 degrees north, right about where this line is, we see a change from one sort of compositional fingerprint to another compositional fingerprint, where you see this red compound in red, which is called homerine. I've mentioned it once before, it becomes very abundant. Homerine is a molecule we've never measured in the ocean before, and it turns out that it's one of the most abundant compounds in terms of the metabolites that we found. We've done this same cruise 3 times now. So that was the first time, and this data now is from the second time. So I'm mixing cruises, but I just want to illustrate with this because we have additional data from this cruise that we didn't have for that first cruise. We measured the metabolomes as we went north, just as we did before, and you can see the concentration of metabolites increasing as we went north, and that corresponds really nicely with an increase in the phytoplankton biomass as we go north. Each of these is a type of phytoplankton that we were able to measure in situ in the ocean. And it's very reassuring that these metabolites actually track the total biomass that we're measuring, because if they didn't, we would be a little concerned. So it's nice ground truthing. And what I can say is that in this gyre area, the organisms that dominate the phytoplankton community are the cyanobacteria called Prochlorococcus and pico-eukaryotes. And as you cruise north, this community really changes a lot. The Prochlorococcus die off, and then we get a different cyanobacteria called Synechococcus, along with a big increase in this pico-eukarya. That just means small eukaryotic phytoplankton, and then a bunch of other organisms like diatoms and haptophytes that are present in the further north stations. So when we look at the composition, or actually, first I'll show you the overall amount of the carbon that we can account for with metabolites is somewhere between 2 and 10% of the carbon in the particles. That doesn't mean that the cells aren't excreting larger amounts. This is what they're holding inside their cell at this given moment when we sampled. So we were really interested in being able to connect the metabolomes that we were seeing with the phytoplankton species that we were seeing, and wondering whether this taxonomy of the phytoplankton actually looks like the metabolome that we're seeing, and can we trace these compounds to specific organisms or organism types. So to investigate that question, a bunch of my lab members, as well as Bryn Durham from Ginger Armbrust's lab, grew up 21 phytoplankton species and 15 bacteria species, which I'm not going to show you that data for, but the phytoplankton are pictured here. We grew them up and measured their metabolomes, and these are the top 20, or I think 20 compounds or 16 compounds that we could quantify once again in cultures, and these are growing exponentially, very happy with lots of nutrients. And you can see that the different species look really different from each other, and we have different types now. We have cyanobacteria, diatoms, dinoflagellates, haptophytes, and green algae. So those are all components of our community in our stations in the North Pacific. And in addition to those, I also have this ammonia oxidizing archaea here on the very left, which has a very unique composition to its metabolome. But you can see in multivariate space that that metabolomes, I guess, recapitulate the taxonomy that we have here. And so you can see that the dinoflagellates kind of group together, the diatoms group together, and so on. And so that's because each of these species looks a little bit different, but they look more like their own type than other types, right? Cyanobacteria look more like each other than they look like diatoms, and so on. So this is a pretty powerful tool that we can see these differences. And so we went back to our environment, to this cruise that I've already mentioned, and we didn't just measure 20 molecules, we measured 350, and we don't know what all of them are, but we know what a bunch of them are. So we have 350 high quality mass features that we mined out of our untargeted metabolomes from this environment. And we looked for those same exact 350 metabolites in the 21 phytoplankton cultures that we grew. This is just the names of the 21 phytoplankton cultures. And when we did that, I just want to show you the 350 metabolites. The ones that we know their identity are in black, the ones we don't know their identity are in gray, and this is the retention time on the column that we analyzed them on, and the mass of the ion that we detected. So you can see these molecules are somewhere between 50 Daltons and 800 Daltons. And if I take one of those unknown molecules right here, this gray one right here, and I look at its trend across this transect in the ocean that we cruised along, you can see that this particular molecular feature is high in the gyre and decreases as you go north. We don't know what it is, but it has a pattern, and it's probably a metabolite, and we'd love to know what it is. It has a mass of 160. So you get the idea. So each of these lines across this figure are one of those metabolites that I just showed you the trend for. And there's 350 of them. And then each of these is a station south, or well, it's 23 north to 37 north. So that's our data from the environment. And you can see we clustered it, Catherine clustered it, and she found that there are groups of compounds. Some increase as you go north, some have no particular pattern. Some are really highest at this 33 to 35 degrees north, and some are really highest in the south. So we did the same thing. We looked for these 350 molecules in the cultures, and we clustered them. Catherine, again, clustered them. And they fall into these clusters that I'm showing you. And we have named these. This group here is the core metabolome. It's in pretty much everything. This one is sort of a mixed bag. This group is primarily found in the cyanobacteria. This group is primarily found in diatoms, dinoflagellates, and haptophytes. And then we have a bunch that were rarely observed in our metabolomes, and some that were actually completely absent from our cultures, a bunch. So we have these two clusters, one from the environment, one from cultures. How do they relate to each other? Well, here is these black lines are showing you places where the metabolites in this cluster from the ocean are more than randomly associated with this cluster from the cultures. And so it's saying that we can actually think of these to some extent as taxonomic indicators, right? So I'm going to give you an example down here. This cluster is highest at 33 degrees north. And if I go over here with this black line, it's showing me that these compounds are really enriched in diatoms, right? So we think that that's probably a place where we would find diatoms growing. Did I say that right? I said dinoflagellates, that's what I meant to say. Okay, so if I take a specific example, this is a molecular feature. This is the pattern of the molecular features in this cluster C, and I'm going to pull one of them out where the star is, and it happens to be on this black line right here, and I'm going to trace that black line up to the culture, and here it is. It's in the cyanobacteria cluster. What is that compound? We happen to know the identity of that one. It's glucosylglycerol. This molecule right here. It's used as an osmolyte, primarily by Synechococcus, one of our cyanobacteria. So if we look at the trend of Synechococcus along our transect, we can see that it's pretty low, and then it peaks in this mid-latitude and goes back down again. And that's exactly the trend of this molecule as we go from south to north. So we can pretty much say that most of this glucosylglycerol is actually coming from a Synechococcus, which is pretty remarkable. So what this is showing us is that as we cruise from Aloha into the northern waters, the biochemical composition of the phytoplankton or the whole community reflects the phytoplankton present, and we can actually predict to some extent who is there based on the metabolites that we see. And each of these structures has a different biochemical function, and they all have different biogeochemical functions, meaning they have a different role in the community as well as in the organism that made it. So that's what I'm calling the chemotaxonomy aspect of our metabolomics. And now I want to show you, those were sort of snapshots in time of the metabolomes from the environment and from cultures. And what I want to talk to you a little bit now about is how flexible these molecules are inside cells. And we have a bunch of projects going on with this, but I'm just going to tell you about one of them, this diel study that we did. So we went to, I meant to put a map on here, but I forgot. It's just Station Aloha right near Hawaii. So we are in the middle of the gyre, right next to Hawaii, about 100 miles from Hawaii. And we monitored in situ in real time the particulate organic carbon and the biomass of these different components of the microbial community, these picoeukaryotes, Prochlorococcus, Crocosphera, Synechococcus. This is data from Ginger Armbrust's lab, who uses a flow cytometer, in situ flow cytometer, to in real time measure the abundance and biomass of all of these organisms. Angel White has an in situ detector for particulate organic carbon, and that's her data up here. And what we can see from these signals is that biomass of all of these groups and of the total particulate organic carbon pool oscillates on a daily cycle. So they generally tend to go up during the day and come down at night. They go up again during the day when the sun is shining, and they come down at night, which makes perfect sense for a photoautotroph dominated community. We measure metabolomes every 4 hours along this time course, and what you can see is that in the morning at 6 a.m. metabolites are less abundant than they are at 6 p.m. And you might say, well, that's just because the biomass increased. But actually, when we look at the percent of the particulate organic carbon that we can account for with metabolites, it actually also increases by a significant margin. So we think that the cells actually have a higher concentration of metabolites within them when they get to the end of the day. They spent the whole day with the sun shining, biosynthesizing and making organic carbon, and their cells are chock full of metabolites. This figure just shows you the metabolites and the time of day during at which they peaked. So they could have peaked, this 1800 to 6 p.m. So that's all these compounds peaked at 6 p.m. And then this on the y-axis is a fold change. So these compounds, the biggest fold change was 12-fold change, which was trihalose. But I've also plotted, or Angie also plotted, this particulate carbon and other members of the phytoplankton community to show you how much they oscillated. So they oscillated somewhere around 2-fold per day, and many of our metabolites, as I said, oscillate with much greater oscillations. So these oscillations, like I said, are indicating that metabolites are higher concentration in the afternoon. And it kind of makes sense if you think about it. Enzymes, if they're going to be high rates of enzyme activity require high substrate concentrations. And so the more you have in your cell, the faster the enzymes can work if they're not saturated. So it suggests a high level of biochemical activity during the time when the sun is shining. We saw oscillations in compounds that are sort of universal to all life, like the methionine cycle compounds are pretty universal to all life forms. And you can see that don't worry about the names again, but these are compounds that are in everything and they are oscillating. So the entire community is oscillating. And then we also have compounds that are fairly specific to specific organisms. Based on our culture collection, we know this, and that are also oscillating. So it seems like a synchrony with the whole community as well as within some individuals with these compounds oscillating. So I'm going to tell you a couple of vignettes about a couple of these compounds, trihalose, and then this DHPS and isothionic acid. Trihalose is interesting. It's considered to be an osmolyte. It helps cells deal with stress. In higher plants on land, it helps them deal with desiccation and those kinds of stressors. In the ocean, it's more about salinity, we think. But this compound had the biggest oscillation. It had a 12-fold change from dawn to dusk. And we used transcriptomics and genomics to discover that we think that pretty much the only organism out there who's making this molecule, I mean, there's others, but mostly it's a cyanobacteria. That's a nitrogen-fixing cyanobacteria called Crocosphera. And they're probably building this compound up during the day and consuming it at night as an energy source, but also to draw down intracellular oxygen concentrations because the protein that they use to fix nitrogen is oxygen sensitive. So this is a way for Crocosphera to store enough energy so that they can actually fix nitrogen, which they do at night. And then there's no sun shining at night to fuel that, so they have to actually store that sun's energy, and they do it partially in the form of trihalose. Another group of compounds that are also considered to be osmolytes are the sulfonates, and Bryn Durham is really focused on these compounds. She found that these compounds had pretty big oscillations, and in our culture collection, they're mostly associated with eukaryotic phytoplankton, mostly the diatoms. And there's several possibilities for why these compounds oscillate with the Crocosphera. I suggested that the Crocosphera itself was consuming the trihalose. And I think by analogy, I'm tempted to say that these phytoplankton are also consuming their own DHPS and isothionate, these small molecules. The other possibility is that they're just excreting them at the end of the day and making them available to the heterotrophic community as food. So we don't really know which one was happening, but we did see genes for that are the genes used by heterotrophic bacteria to consume these molecules. And you can see that they get converted into things like pyruvate and acetyl-CoA, which are TCA cycle compounds that suggest that they're using these to make energy as well as to create biosynthetic intermediates to then make other things. But it also could be that they're being converted into glycine, which is an amino acid that they could use to make protein. So heterotrophs are definitely consuming these molecules, but we don't know. But I think, like I said, based on analogy, that this large swing over the day-night cycle is probably in part because the phytoplankton are actually using these compounds, again, as an energy storage compound. They're really readily available, don't have to be, they're not, big, like starch is a big molecule. It has to be hydrolyzed and then finally consumed through glycolysis and then finally put into the TCA cycle. It's a much longer pathway. These are really readily available molecules. As soon as the lights go out, they can use these compounds for energy. So that's what I'm going to say about physiology. We've done a whole lot of other experiments showing how these molecules change over different environmental conditions, and there's many, but that's the one I'm going to tell you about. And then finally, I have a couple stories about the fate, and I guess it's 3:40, so I might go a little quickly through this, but we're looking at where these molecules go, who's using them, and how. And one of the things that we want to do in order to really tackle this question is to look at the compounds in the dissolved pool. And so Josh Sachs in my group, as I mentioned, he's the astrobiology student in my group, and he has been working on this method to perfect this method for measuring dissolved metabolites. And here's the first data that we have. He measured a few different samples, the Ocean Gyre, the Station Aloha, then the North Pacific, Puget Sound, and Lake Washington. Some local highlights. And some of these compounds have never been measured before in the dissolved phase, including the most abundant one, homarine. And if you look at the composition of these different environments, you can see this is now not the total abundance, but the relative composition, relative abundance of each one. And you can see that the composition changes between these environments. And it doesn't look exactly like the particles, but it looks somewhat similar. There's probably been a lot of editing, either through excretion, selective excretion, or through selective uptake by heterotrophic bacteria. But I want to focus on homarine again, because it is so abundant in the North Pacific and also in the Puget Sound in the dissolved pool, as well as the particulate pool. In our culture collection, homarine appeared to come primarily from a couple of diatoms, but mainly this cyanobacteria called Synechococcus, where it reached 400 millimolar inside the cell, which is really high. In our environment, you can see that as you go along the gradient, this homarine increases as you go north, just like our Synechococcus peaks here, and it also has a peak there, but it then continues to go up, probably because their eukaryotic biomass further to the north is also making homarine. We were interested in the availability of this molecule as a food source for heterotrophic bacteria, and so Catherine grew this heterotrophic bacteria called Gregaria pomeroy on several different substrates, one of which was homarine, which is in green here. And so over time in this incubation where we were growing this bacteria, homarine resulted in the least amount of biomass production. Other molecules like taurine, trigonylene, acetate, and betaine all resulted in a much larger amount of biomass being created in these incubation and this culture. And so it doesn't seem like homarine is a very good food source for this particular bacteria. And that's not terribly surprising that there's some specificity, because homarine is known as an osmolite, but it's also known as a morphogen, a methyl donor. It's used in predator-prey detection. It's being researched as an anti-fouling agent, and it's also toxic to butterfly fish. So there's a lot of different ways this molecule is acting in the environment. And we don't know the genes responsible for biosynthesis or catabolism. They're not annotated in KEGG pathways. So we can't really find the genes for this in the environment. If we grow another Rosiobacter with its normal food source is in blue here, it's growing over time very nicely. If you add homarine to that, it delays its growth. So it seems to eventually grow to pretty high abundance, but it's delayed. And if you add enough homarine up to 12 molar, you actually this organism ceases to grow. So there seems to be some very interesting biochemistry related to this compound. We're very curious to search for who might actually like to consume homarine, because we know that somebody does, or else the ocean would be full of homarine. So we're searching for that. Another molecule that is more abundant, or is also very abundant, is called glycine betaine. This has been called the most abundant osmolite on Earth. Also has many functions. And this one is one that Angie Boysen was very interested in as a nitrogen source, and also as a carbon and energy source to the heterotrophic bacterial community. So she did an experiment at sea where she took seawater and added isotope labeled glycine betaine to these incubations, and then waited and measured the uptake of glycine betaine, isotope labeled glycine betaine, into the community. So this is the community in the northern station. This is pretty far north into the North Pacific at 41 north, and this is further south at 37 north. And I put little stars in our profile here in our transect to show you where we measured those. And it looks like the southern community at 37 north right here is more primed to take up the glycine betaine. The enzymes seem to have higher affinity and are actually taking up that molecule much more quickly than in the northern station. And we think that that's probably because the amount of inorganic nitrogen, like nitrate and ammonia, were very low at this station. It's almost unmeasurable. And as you go further north, there's plenty of nitrogen available for the community. So they don't really need a nitrogen source like this. They can just use inorganic nitrogen. Angie was also interested in knowing where that glycine betaine was going once it got into the cells. So the majority of it remained as glycine betaine in both north and south stations. This is, as you get more yellow, you get higher concentrations of the labeled glycine betaine within the cells. But it didn't all remain as glycine betaine. Some of it got turned into other molecules like sarcosine and these other ones that I've named here. I don't want to get too much into the weeds of the details of these compounds, but just to show you this very complicated figure, glycine betaine has very well-known pathways of metabolism. It can be, for example, demethylated through this pathway, and which can produce glycine, which is an amino acid used in making proteins. It can be, this trimethylamine can be removed, and that can be converted to ammonia and formaldehyde, which has now been basically completely remineralized to nutrients again. And it can also be put into lipids. or it can be converted through this other pathway through carnitine. So we saw evidence that there was a difference in the north and the south as to which pathways were favored. And in the southern stations, it favored sarcosine, this pathway to sarcosine, which might mean that it's actually retaining this organic molecule as an organic molecule and maybe making things like glycine to produce proteins because it wants to retain that nitrogen, whereas in these other places, it is possibly in the further north station is possibly being converted to TMAO, which is also an osmolite, but then finally to ammonia and being just spat out as ammonia because they don't really need that nitrogen, that community, perhaps. When we look in into Josh's data, we can see that in the more northern stations and in the Puget Sound, we see a really high concentration of this TMAO compound right here, suggesting that in these nitrogen-rich environments, this TMAO is not just within the cells, but is actually also in the dissolved metabolite pool, which is pretty cool to see that it's higher at these stations where that pathway seems to be favored. So our data is self-consistent, which is really nice. We still don't really know who's taking up this material. So we don't know this glycine betaine that's being held as glycine betaine could be taken up by heterotrophic bacteria, but it could also be taken up by phytoplankton and used as an osmolite by the phytoplankton themselves. That means that they can use it and they don't have to make their own glycine betaine. So it's a way of acquiring a nitrogen-containing organic molecule that has important functions in the cell without having to biosynthesize it themselves. So with that, I'll conclude. And just to say that I hope I've given you some insight into how metabolites can tell us about the sources of organic matter in this chemotaxonomy section of the talk, how the metabolite pool is quite flexible and reflects the physiology of the cells and the environment that they're living in, and that we can look at the fate of organic molecules and that actually that different communities process these organic molecules very differently, and they take them up at different rates. And so these are the kinds of interactions that we're interested in studying, and we'll bring this kinds of things to the lab to investigate more fully. And just to make a note that, that we've studied a lot of organisms and a lot of a pretty big, one of the big oceans of Earth, but there's many more microbes to study, many more structures and functions to identify, and also many more oceans and ocean worlds to study. So maybe someday some of these tools will be applied to other ocean worlds, and we can look for some of these very small polar compounds that might be indicators of life or precursors to life. And with that, I'd thank my lab group. They're looking a little happier on this Zoom call. Sometimes we actually joke around a little bit. It's hard, and also some of my collaborators from the Simons Foundation collaboration that I'm involved in called SCOPE, and NSF as well. So thank you very much. Is Roger frozen? Oh.
Okay. Thank you, Anitra. Does anyone have any questions for Anitra? You can either raise a hand and we'll try and get to you, or type a question into the chat. I can call.
Was there a question? I did not hear one. I think Roger might be frozen.
I think that, this is Sarah. Hi, Anitra. I think that Roger's email is, his internet connection is having difficulty. It looks like the chat window has a lot of great questions for you already. So if you want to go ahead and drive, I bet that Roger would be grateful.
Okay, so I see, all right, I'm getting the chat open and I can see, do you investigate the effects of plastic particles on these metabolites? We have not. I guess that's a simple answer. We have not. I do know people who are investigating the fact that plastics can influence the bacterial community, and presumably that would mean that they would impact the metabolite composition, but we have not measured plastics, and yeah, so we don't know about that. Let's see. Wondering about the extent some of these compounds can be considered biomarkers, or if any of them could be produced through abiotic means. I'm not really an abiotic primordial soup kind of a person, but I guess I would bet that yes, some of them can be with the right conditions for sure. Yeah. I don't have a good answer to that though. That's really specific. Yeah. And I think that's the only questions. Oh, wait. Oh, Brooke, you have a question.
I can actually verbalize it if you want.
All right, great. Good. Someone else will talk.
Sorry. Great talk, Anitra.
Thank you.
I'm hearing a lot of crosstalk. So anyways, yeah, so with regards to, you might have mentioned this and I apologize. Is it the intracellular or extracellular metabolites that you're predominantly focused on?
We have so far been predominantly focused on the ones inside the cells, and almost everything I showed you was that, except for this last, the last data, which was from Josh. You know, this last bit right here. This is the only dissolved data I showed you.
Okay. And do you think that you can take dissolved data and kind of Fourier transform, pull out specific signals? Is that potentially the goal? Like specific signals from diatoms or dinoflagellates or?
I mean, I'm really interested in the extent to which, you know, we can put one up next to the other and say, how similar are they? In the one place where we have like compared the homarine concentration inside the cells in a liter of water is exactly the same as the amount of homarine dissolved in the water in 1 liter of water, which seems very coincidental, but it was the case both at Aloha in the Gyre and also in the North Pacific. So I have no idea if other compounds will show the same sort of relative, you know, abundance, but that would be, I'm interested at a first glance to compare, you know, how similar do they look, right? Or does the extracellular metabolome indicate a really large degree of editing of that, either through selective excretion or selective uptake? You know, the yummiest molecules will disappear first.
Right. If nobody else has, I have one other question. This
Sure.
is my question that I asked Neil deGrasse Tyson, and he totally missed it. I mean, there's no answer. If you had to pick a metabolite to hunt for on another planet or non-Earth planetary body, what would that be and why?
I think I would probably pick a betaine, one of the betaines like glycine betaine or a simple one like that. Yeah, because yeah, that's what I think.
Can you pull up the structure? I guess I ask because, like, I mean, we fight with this with regards to the stuff we're looking at. And to remind people which haven't seen me speak in a while, we're looking at cold active organisms and salinity specific or cold specific proteomes to understand if there's specific folding aspects of these proteins that might indicate that that protein is a salinity marker or a cold active marker that might be present on another world. And it comes down to trying to figure out, I don't remember who asked the question, maybe it's Rika asking about whether or not it was abiotic or biologically made. And I know that there's discussions about how complex a molecule has to be in order to, like, there's, 5 steps. I think it's 6 steps of synthesis. Speak up if somebody remembers that exact number. But in order to be considered biologically made.
Oh.
And whether or not betaine makes that mark or not.
Wow. I don't know. Someone else might be able to answer that better than I can. I will say, in terms of the cold adapter, the homarine, it does increase as you go north, and it's even higher in the poles. Like Hannah Dawson works with my group. She's one of Jody Young's students, and we work with her on polar diatoms, and there's just a ton of homarine in those polar diatoms. So that might be a good one to look for, or something like it. I don't know what it's doing. I have no idea. It's named homarine because it's Homarus is lobster. It was first found in lobster, which is also a cold dwelling organism, right? It doesn't like to live below whatever temperature. I don't know which temperature, but New York about in the ocean. So that's an interesting question. I mean, there's lots of really small molecules that are obviously biological that don't have that many steps for synthesis. But if you wanted to be sure it was biological, then I guess you're saying you need more. Is that what you were saying?
Yes. Yeah. Well, I looked for like a calculator to figure out like how many, alkyl groups and OHs and need to be added that are comes down to non-similarity in the actual structure.
Mm-hmm.
But I'm more or less hand waving because I haven't read all those papers recently.
Yeah. Cool. Yeah, I might look for one of the betanes. I might also look for a sulfonate. I don't know what, I'm ignorant enough. I don't know what atoms are most abundant on these other planets. Like, you know, I'm not sure that matters exactly, or you know, whether we know what kinds of salts or raw materials are available in these other ocean worlds. You know, is it mostly sulfur? Like in Earth's ocean, we have a lot more sulfur than nitrogen. So you might expect sulfur containing compounds like a sulfonate. But the betanes are like the best osmolytes and very efficient. So I don't know.
Are there any more questions for Anitra?
I think there might have been some more down in the chat if you scroll down.
It's quite a comment about silica as a food source. So yeah, that's because diatoms make their cell wall out of glass, which is silicon dioxide. So that's why silica is a nutrient for some organisms. I think it's required by a lot of organisms, but they have a very large requirement for it. I don't see any others in my chat.
Okay, if we're all out of questions, it's been just about 4 o'clock. I think we'll leave it there, and unless anyone has a particularly urgent question, you can always contact Anitra afterwards if you do have a question that you think about later. But with that, I'd like to thank Anitra very much for giving a talk that mixed biology with earth and ocean science and went to a great deal of trouble to try and link it to other planetary bodies as well, which covers the whole range of astrobiology subdisciplines. So thank you very much, Anitra.
Thanks to everyone for coming out right before the holiday.